Are ERVs evidence for evolution? I've heard over and over again that ERVs are just some of the best evidence for evolution. And here's an example of a clip from a popular YouTube video stating that case.
In contrast, if we want to believe the fixed species view, we're forced to conclude that these viruses are simply shared by coincidence. When we do the math, even making sure to account for the 9 viruses not shared by the 2 species, the chance of this happening by coincidence is less than 1 in this crazy number right here. This evidence should be enough for even the most reluctant yet rational person to carefully set aside the fixed species view. Endogenous retrovirus DNA alone is more than enough to independently confirm what we already knew from the study of fossils.
So, pretty strong words, and it's convincing a lot of people. Well, I'm here with biophysicist Cornelius Hunter, and he has a lot to share about this. So, welcome, Dr. Hunter. Thanks for being here.
Thank you, Rebecca. I'm delighted to be here. And that was a very highly produced video, and it's convincing people. Some top scientists were involved in making the video also. And in that underlying calculation that you saw, very impressive, a lot of zeros there. The chances of the fixed species view, as he said there, is 1 in a 1 followed by thousands and thousands of zeros. So the chances of the fixed species view is essentially 0. And so that leaves evolution and common descent as the obvious explanation. Now, can you bring up my slides? I'm going to say a few more words about this video, but that calculation, that argument is common. It's not just in that video, but that's a good example of that argument. And it depends on certain assumptions. And so mathematics and science are only as good as the underlying assumptions. And if those underlying assumptions are false, then there's a problem. So first, I want to explain that calculation a little bit more. So imagine you're looking at a genome of an organism, and in that genome, there's a virus. That red dot represents a virus. And you look at another organism. So maybe that first organism was a chimpanzee, and this next one is a gorilla. And then you look at an orangutan, and then you look at a gibbon, and you look at the human, and so forth and so on. And they all have this virus in the genome at the same place, and it's the same virus, the same type of virus in the same location. Now, there are millions and millions of locations in the genome where these viruses can lodge. Now, by the way, viruses that can insert a copy into the genome, those are called retroviruses. So that's what we're talking about, retroviruses. And when usually they get into your nose and your lungs and your nasal tract and so forth. That's what we think about with viruses. But they can get into your sex cells. And then when you reproduce and you have children, they can get in via the sex cells, passed on to the next generation. That way they become, they can overtake the population. They can spread in the population. And geneticists call that the fixation. They get fixed in the population. So that's what we're talking about here. Are viruses that interjected, they integrated into the genome in the sex cells and then spread through the population. And oftentimes they are inactivated by the defense mechanisms of our bodies. So they're not necessarily a problem, but they're there. So those are called endogenous once they have done that, once they've gotten into the population. So they're no longer coming in from the outside. They're not exogenous, they're endogenous. So just some terminology so we're straight. That's what ERV means, endogenous retroviruses. So again, imagine you're looking at several species and you see the same virus in the same location, same type of virus in the same location of multiple species. So is that a coincidence? Well, probably not. That's what this video is arguing. That's what this common argument is arguing. it really can't be a coincidence. It's just so unlikely because there's millions and millions of locations in the genome where this virus can insert, it can integrate itself. And so here are the key premises, and the logic is very simple, actually. ERVs are genomic junk, typically. They are found in the same location in different species, the same genomic location in different species. They form a common descent pattern. There's no reason for this because ERV location is random. And therefore, the conclusion is it cannot be via what's called convergent evolution. It couldn't have happened just over and over and over again independently, according to a model like this. So you have these different species, and these red dots here represent the integration events for each species. So it just happened, independently for each species, and then it just happened to go to the same genomic location. That's just too unlikely. So instead, you look at they're arguing for this evolutionary common descent pattern where, okay, it didn't happen once for each species, it happened once prior to the common ancestor of all the species. And that makes a lot more sense for them. So that's the idea here. Or if you look at that, they're going to zoom in on the chimp and the human. And so here's the convergent model for the chimp and the human. It happens independently twice versus once before the common ancestor. Now, the virus itself consists of a couple different parts. You have its genome in the middle, and then you have some flanking regulatory sequences that are shorter on the ends. When it integrates into the genome, again, you have the coding sequences, but the flanking sequences have been processed, and they're called long terminal repeats, LTRs. That integration process, keep this in mind. I'll come back to this in a second because that integration process is important. That processing of the LTRs is going to be important. Now, this is called the provirus. Once it's integrated into the genome, it's then called a provirus. Just a little terminology, not critical. So there's your long terminal repeat. Those are those end caps, those flanking sequences in Wikipedia. Now, it turns out that the vast majority of the retroviruses, the endogenous retroviruses in our genome, are not that whole thing there. It's actually just the LTR. So that's an interesting twist. All of a sudden, you don't have the full thing represented typically. More on that in a second. So these are the key premises, the key logic we need to keep in mind. The calculation and that number are only as good as these key premises. ERVs are genomic junk. They're found in the same location. They're forming a common descent pattern. There's no reason for that. And so therefore it cannot be via common descent. We're going to find out very shortly here. These are all false in important ways. So I'm going to go through this outline to explain that. It's actually very interesting. This opens up a lot of interesting questions. An interesting example of how they don't fit the common descent pattern. There often is a mismatch between the proviral body, that genome, the provirus, and the LTRs. And by mismatch, what do I mean? There are different families of ERVs, and sometimes you compare them. And in this case, and from this paper, they're comparing two different families where that genome is very similar between the two different provirus family types. So it looks like they're almost in the same family, or at least they have a common ancestor. They're very closely related. But the LTRs, the long terminal repeat flanking sequences, are not. They're very different. That just makes no sense. So that's just one kind of weird, wait a minute, what's going on here? So a lot of cases like that, where all of a sudden the story starts to break down. Their LTR sequences are very different. This is not kind of a lone example. This is actually something that's observed a lot, and it just doesn't compute on the common descent evolutionary view. Don't worry more about this. I'm not going to come back to this. This is just a kind of an interesting example of what the story is a little more complex than the video is telling us, than this argument is telling us. How about the actual data that the ERV that the video was using? What about these 210 ERVs? So here's a snapshot of the data that they were using in a spreadsheet, and they are looking at these different species. There's the human species, then there's chimpanzee, gorilla, orangutan, gibbon, and rhesus, and that's interesting. Now, if you focus now, so each row is one of these 210 integration instances where this family, this type of ERV was integrated into the genome. If you look at the gibbon, so in the first few rows, they're all in the first chromosome, which is they need to be in the same place. Remember, so okay, it's giving the coordinates, the actual locations in the genomes, and they're all in the first chromosome. So okay, good, that makes sense, right? Oh, wait a minute, you get to the gibbon, and it's not in the first chromosome; it's in the 12th, and then there's a couple that are in the 5th. And what's going on there? Why is it in a different chromosome in the gibbon? Is that actually a common descent pattern? It doesn't look like it. Well, there was the explanation is there was genomic rearrangement in the gibbon. So you had this, once it split off into the lineage of the gibbon, things happened. So things got rearranged. Okay, maybe that's true. It's probably not a showstopper, but kind of strange. It does happen. What about some of these orangutan coordinates are pretty smaller than the others. Oh, well, those are just the long terminal repeats in those cases. Okay, it's possible. It's just, but again, it's a little more complicated. What about this one here? This is a particular integration event where you notice these X's in the spreadsheet. What do those mean? The X's mean there isn't one. So all of a sudden, they're not all in a common descent pattern. There are missing ERVs in certain species, certain instances. They're not there. And in this case, they're not. There's 3 of them that are not there. The chimpanzee, gorilla, and gibbon don't have the ERV present. What's going on there? Well, the explanation is they got deleted. So one of the interesting things about these ERVs is that they are retrotransposons, meaning or jumping genes. Transposable elements is more generally referred to. So they can move around. They can be deleted. They can have a copy and paste and so forth. So there's a bunch of different mechanisms by which they can move around. They can have reinfections from the outside world, or you can have them moving around in the genome. And so all of a sudden, you can explain a wide range of outcomes. And you can have outcomes like this, which the evolutionists are not bothered by, where it doesn't fit a common descent pattern. Because, they say in that, and they do say that in this case, it was deleted in the chimpanzee, gorilla, and gibbon. And so it was inserted prior to the rhesus monkey, and it stayed in the rhesus and the orangutan and the human, but it was deleted in the other 3. Well, what are the chances of that happening? That seems like kind of unlikely. Well, you can actually calculate that, and I'll show the calculation here. But here's the explanation. Okay, these red X's represent deletion events. So you have 3 deletion events, according to evolutionary theory. So what are the chances of that? So you can calculate that. I'm showing that here. I won't go through the calculation, but you can pause and look at the numbers and see how I got the number. But you have a probability of deletion for the chimpanzee, gorilla, and gibbon, and then you can use that to calculate the overall probability of this happening in a single instance, a single integration event. It's 0.0029. In other words, about 3 in 1,000. The chances are about 3 in 1,000 of that happening. So what's going on there? That seems like not a very good theory here. Well, you can argue, the evolutionists can make arguments about that. Maybe there was, this ERV is farther out on the chromosome. It's farther from the centromere. And maybe deletion events are more common farther out from the centromere. There's always an explanation, right? So maybe not a showstopper, but again, the video and the argument doesn't give you these sorts of insight, doesn't tell you these things. Suddenly things start to get more complicated. There's also a major problem when you drop this assumption. Remember, the key assumption here is they fall into a common descent pattern. That's what they do. ERVs do what ERVs do. They fall into they show up at the exact same place, and it's a common descent pattern, except when they don't. And that change, that forfeiture is huge. That's an enormous change in the argument. When you do that, you're no longer predicting. Your theory doesn't predict this. You're okay if it didn't happen. In fact, as I said earlier, there are hundreds of thousands of ERVs that don't fall into this pattern. Evolution is perfectly okay with that. Philosophically, you have forfeited the entire argument. You could take this spreadsheet and you could put more X's in there, and the evolutionists would say, fine, no problem. You could put more X's, no problem. So in other words, evolution can explain any outcome. And so it can't claim any one particular outcome as a slam dunk confirmation proof of its theory. So very important forfeiture there on that assumption. Okay, so it's not very likely that particular one. Now move on to the next point here. ERV integration sites are not random. So remember, that was a claim that they're just random. When ERVs integrate into the genome, there's all kinds of correlations. They're not even across the chromosomes. They're not even across the location within a chromosome. They tend to be closer to the centromere. They correlate with whether there's genes in that area or not. And where there are, if it does land close to genes, they correlate to whether it's an intron or whether it's a regulatory sequence and so forth. Transcriptional activity has an impact. Orientation, which thread of the DNA, which coil of the double helix is it on? By GC content, et cetera, et cetera, et cetera. So there's all kinds of factors that seem to correlate, seem to influence whether a ERV is going to integrate. It isn't just a random process. Here's an interesting example. This study observed integration events. They actually observed them. So this is a great study. They synthesized an ERV, a consensus ERV, and then they, in the laboratory, had it go in and infect cells and measured where is it infecting. And it was, really, really good study. And they were not random. They didn't just randomly integrate. I'm not going to go into the details because there's a whole bunch of correlates, but they weren't just randomly integrating. This is
okay. So basically you're saying like there might be 10 million different sites, but it doesn't mean that it's going to be drawn to that site. It may have a particular site that it's drawn to. And so there may be a correlation, like in different species, it could still insert in the same site.
Exactly. But hold that thought, because I'm going to come back in just 1 second, because it gets a little more interesting than even that. But that's the point, in that the assumption was they have these key premises. One of the key premises was it's random. There's just no reason for these things to land in the same site. And if you drop any one of these assumptions is not true, then that number doesn't work. And so this one is not true right off the bat, but it gets more interesting in just a second. Here is another study which says different retroviral genera, that is different types of ERVs, exhibit distinct site preferences. For example, lentiviruses preferentially integrate into actively transcribed genes. Now, lentiviruses are not retroviruses, or at least they have not endogenized yet, but they are retroviruses. They're not. What did I say? Let me make that clear. They're retroviruses, but they have not endogenized, as I understand it. But it's relevant to look at their integration site preferences. And in this paper, they find that they preferentially, or they're noting that lentiviruses preferentially integrate into actively transcribed genes. So granted, this is not endogenous, but it is a retrovirus, and this is very significant. In other words, it has a preference of, there's tens of thousands of genes in the organism, in the cell, in the genome. It has a preference. Only a small fraction are opened up for business and are actually being used and actually being copied and translated. So all of a sudden you've gone from 10 million possible sites down to a much, much smaller number. If it's going to be preferentially integrating at actively transcribed genes, that's a much narrower filter. So again, this isn't for all viruses, but it's very interesting that you do find these significant integration site preferences. Okay, another one of those key assumptions was that the ERV proviruses are junk. Well, no, they're not. They're not useless junk, and this gets very interesting. So remember that I'm repeating this slide here. ERVs are genomic junk. No, they're not. ERVs provide a major driving force in the emergence and diversity of the placenta. Wait, what's the placenta? What's this about now? Placenta. The placenta is that thing in the mammalian reproduction system that helps, the womb and everything, right? That's kind of the device that the newborn is living off of, and it's the connection device to the mother. A very important design, very important structure for reproduction. The ERVs, there are regulatory sequences that look like they are associated with ERVs or derived from ERVs that are a major driving force in the creation of placenta. When a placenta is created, it's relying on these genomic sequences.
Can I interrupt really quick? Because this is kind of a pressing question related to this. When I read about the ERV that allows for the placenta to attach or whatever, then it kind of made me say, well, are these even really what they're saying they are? Are they really endogenous retroviruses? Because right. Do you have an answer to that? Like, is that in question?
It is a question for sure. What exactly is this? Because what happens is you run into a serendipity problem. So under evolution, you have to imagine that these viruses evolved to multiply. That's all they do. They don't do anything. They're leeches, right? They're not even considered to be life. They're just little machines that just multiply. And so I'm going to talk about this in a minute, where you just have a virus that evolves to multiply. And then all of a sudden it is a crucial component for further evolution. So that's serendipity. So the theory is now, because of these findings, the theory has enormous serendipity, and it's not good. Serendipity is not good in scientific theories. So I'll talk about that in a minute. So yeah, whether these are actual, how those sequences got there and why they're doing what they're doing is an open question. But no, I'm not going to have any magic answers about what they are. I'm analyzing this argument, and the argument relies on enormous serendipity and false premises, and that's what we'll see. But to your larger question, Rebecca, yes, that's a very interesting question. What is going on here? And that's a hard question. See, all of this whole ERV area is a current, very active area of research, and so there's a lot of unknowns. And papers will say this. I have quotes, from that. Authors saying, yeah, we still don't really understand this. This has only been looking at this for the past few decades, and a lot is every year we're learning more things. Evolutionary arguments and strong evolutionary arguments typically are relying on this ignorance, because they will claim certain things about evidence, and then in later years it's discovered, oh, it didn't work that way, and it's no longer evidence for evolution. So good question. But this paper explains that ERVs provide a major driving force in the emergence and diversity of the placenta, and contribute to the remarkable diversity of placental structures across multiple lineages. Now, hold that thought. We'll come back to that. So you have independent ERVs contributed to the placental morphogenesis. That's when they're created. Notice the title of this paper, Friendly Viruses. They're very friendly because they're actually helping evolution along and creating incredible structures. ERV proteins derived from that ERV genome, have been domesticated independently on multiple occasions for the placental functions of several mammalian lineages, thus representing a process of convergent evolution.
Wow. Oh my gosh. So actually, they just destroyed their whole argument, because their argument from the ERVs is based on the positioning of these and how it couldn't have happened,
Right,
multiple
exactly.
times. But now they're saying it happened multiple times.
Exactly. So what I wanted to point out was how complex and how serendipitous this is, but there's something altogether on top of that, which is it happened over and over again. According to evolutionary theory, it cannot have happened just once, and I'm going to explain that. Here is a paper about ERVs. Note the title, From Junk DNA to Genomic Treasure. So we saw the last title, Friendly Viruses. Here we have From Junk DNA to Genomic Treasure. ERVs are transformed from junk to essential genomic players, quote, end quote. Another rapidly evolving system with clear signs of ERV involvement is the mammalian placenta, thought to be driven by what? Independent ERV acquisition. The problem here is it doesn't line up with common descent. This whole placenta story doesn't go back to before the common ancestor, and you can't, have it just happen once and then propagate through the tree, the evolutionary tree. Just because of the differences between the species, it had to have happened after the common ancestor, so it have to have happened over and over and over again. And so that's called convergent evolution. These events provide what? Species-specific placental programs in a remarkable example of convergent evolution across all placental mammals. It happened independently. And so you got that, Rebecca, because I want to pound that home. When we went over the key premises and the logic, it cannot be convergence. It cannot be. And now it must be. Multiple acquisition events of ERV proteins in the ENV family group independently in a remarkable example of convergent evolution. So remember that key. I'm going to come back to this slide here over and over again. The key premises and logic. It cannot be via convergent evolution. That was that whole calculation, that big number. It cannot be convergent evolution. Now it must be. So you got that. You got the point there. Another example is the myelin sheath. Well, now what's that all about? A recent and remarkable example of convergent evolution was an ERV essential for myelination. The myelin is the insulating sheath surrounding axons in the central nervous system. So neurons have these long tails. They're called axons. There are electrical signals called action potentials that go through these axons. So you have this long tail. It's going all over your body, sending these electrical signals down your back and your limbs and everything. And so you have these, several feet long axons that have to send these electrical signals. Well, they need a conductor. They need insulation to really work well. Without that, they don't work as well. And so for for the human body, you have to have this. Myelin is the insulating sheath surrounding axons in the central nervous system that allows for rapid transmission of nerve impulses to enable more complex brains. Okay, very important. Surprisingly, evidence suggests that multiple independent species-specific ERV integrations are important for myelination. This is, quote, further evidence that convergent evolution plays a central role in driving genetic innovation. I'm just quoting from these papers. This is my opinion here. So
this
what
is
this
amazing. Okay, I want to just stop and break it down for people who are new to this. So the basic argument, and correct me if I say anything wrong, but the basic argument in that stated clearly video is that, look, we see these patterns where ERVs are in multiple species. This shows that, it would be very, very unlikely, that large number, it would be that unlikely for these same ERVs to hit in the same spot. And so this is like strong evidence for common ancestry. But what we're seeing, sorry, go
let me
ahead.
just interject on the argument. It's powerful evidence against the fixed species view. Therefore, you're left with common ancestry. So it's not a direct argument. It doesn't actually argue for common ancestry directly. It's a 1 in 1 billion, 1 in 1 million that the fixed species view is true. That's the argument.
But then now what we're seeing when we actually look at this is that we do see that the ERVs are coming in, if that's what they are,
Right. Now you get into the whole question of what exactly are these, but from an evolutionary perspective, you have to conclude these are ERVs, and they are integrating independently. These are independent integration events. Now, they don't have to be in the same place in the genome. The simple point is that you have this incredible convergence. You're required to believe in these incredible convergence stories where you're creating these very sophisticated structures. These are neurons, the sheath around our neurons, the myelin sheath, the placenta. There are many more examples that I don't have time to go through. And so, yeah, it is radical, the degree of serendipity that you're calling upon. These things evolved to be a virus, right? Not to be a virus, but that's how they evolved, as a virus. They evolved as a virus that does nothing. It's a leech. That's all it is. It just reproduces. That's all it does. And then all of a sudden, it's critical for life, for evolution to proceed. So evolution is creating evolution. Evolution is creating these things, and then, oh, look what, it would be like, a tricycle. It just happens to be the critical element for a spaceship. A child brings his tricycle to visit his mother, who's working in a factory that builds rockets, and it's, oh, we need this tricycle here. That's exactly what we need to build this rocket. Oh, that was lucky. Your child just happened to bring the tricycle today, and it just works. Now, that's the sort of serendipity that we're talking about. I want to say a few more words about this myelin sheath. So here's a nice picture that gives you an idea of what I was talking about. This red thing on the left is a neuron, a nerve cell, and it's got these tails, which is called an axon, and it needs to have this insulating sheath around it. And that's what this blue thing is. So that's the myelin that's packed around the axon, and that's what the ERV is contributing to. Retroviral endogenization played a prominent role in the emergence of vertebrate myelin. This innovation enabled rapid transmission of nerve impulses that helped create more complex brains and greater morphological diversity. This is how important this is. A critical step in vertebrate evolution. So the serendipity that I've been talking about, evolution creates viruses that just happen to create evolution. So just think of this little sequence here. Evolution of viruses leads to evolution of higher complexity. The way Darwin presented the theory was you just have this kind of linear process of mutations. He didn't think of mutations, but variation biological variations mounting up and accumulating and slowly but surely adding complexity. But here we have something very, very different, much more serendipitous. You have to believe that evolution created viruses, which then became crucial for so much of higher complexity. But there is more, because this had to have happened, over and over and over again. This is the same story. So this myelin sheath didn't just happen in a common, the ERV construction of this myelin sheath, or contribution to the myelin sheath, didn't just happen once before the common ancestor. Again, if you're an evolutionist, because of the differences between these different instances, these different groups of species, you can't say that it just happened once. It had to have happened over and over and over. That's what these red triangles, if you look at these little red triangles, these are the notional look at where this had to have happened in the mammals, the birds, the reptiles, the amphibians, et cetera, et cetera. So again, convergent evolution, it always comes out this way. You have this incredible serendipity, and then, it happened over and over and over again. It just becomes ridiculous. It's not a parsimonious theory. You're carrying an enormous burden. It is not a compelling theory. It could not have been via common descent. It must have been via convergence.
And can you break down for that specific example, what is it that it has to be convergence? Is it because they're in a different place? Is it because they're different? What was it? I'm sorry if I
Yeah, so when you look at them, let's say you look at the mammals,
yeah,
and then you look at the reptiles, and you look at the myelin sheath, and you look at how that's constructed, and what sort of a RNA molecule is key to doing that, and how that maps to an ERV that derives from an ERV. You get differences, like it's a different ERV, or it's the RNA molecule is sufficiently different. There are sufficient differences to where it's, this wasn't the same event.
Okay, so it's actually the construction of the genetic material that it's relying on. It's configured differently.
Yeah.
Okay.
Exactly. So for example, here I'll give you a hypothetical example. I'm not exactly sure what the exact differences were, but I'll give you a hypothetical. It could be mapping to a different family of ERV.
Okay.
Because there's many different families of ERVs.
Mm-hmm.
So maybe the mammals are mapping to one family and the reptiles are mapping to a different family. So you say, it couldn't have been a common ancestor. This didn't happen long ago. That could, that's just a notional example. Bottom line is, it had to have happened independently over and over and over again. So you have multiple problems mounting up here. The serendipity, but then the species-specific convergence, where they said it cannot be convergent evolution. Now, it is two different things here. That when they said it can't be convergent evolution, they're referring to these ERVs landing in the same place. This particular, they're only talking about 210 ERVs, remember, and
Right.
how they land in the same place, but they don't all land in the same place. But their point is, you can't have convergence to explain that. But then later they will turn to convergence where needed with equal or greater unlikeliness. That's the point, that this is a playground for a philosopher of science. They will spend this theory will go down in history for centuries as a case study for the philosophy of science. It is so fascinating how this is spun. Quote, this occurred likely through convergent evolution. Exactly. It is noteworthy
Wow.
that the genes that are required for placental development came from an ERV via independent events across diverse mammalian species, as well as one species of lizard. Another related example of convergent evolution. See, let me explain this passage here. This passage that I just read is from a paper that is not about the placenta. That's not what this paper is about. This paper is about the myelin sheath. Okay, why are they talking about the placenta? They are having to reckon with this serendipity problem and with this convergence problem. And so they're getting it as they're doing this. They're getting it that like this isn't making sense. This is way out of bounds. This is not a parsimonious scientific theory at all. And so they're trying to make sense of it, and they're trying to rationalize it in their own minds, and to help the reader, the reader and the reviewers, the peer review process. So they gotta try and rationalize this somehow. So what do? They point to the placenta and they say, look, it already happened. We already have another case of this. Don't get hung up on this. It happened with the placenta, so it's happening with the myelin sheath. It happens over and over again. So don't get hung up on this. We already have these other remarkable examples. So they admit that it's remarkable, but they rationalize it by, we already know of other examples of this. In other words, if it could happen with a placental development, then it could happen with the myelin sheath. Don't worry about it. ERVs actively shape host evolution through complex mechanisms involving genetic innovation. So this is kind of getting to my point here. Evolution creates ERVs for the purpose, for no purpose at all except reproducing. But then they, actively shape evolution. Evolution creates evolution. Evolution creates further evolution through this very bizarre serendipitous pathway. ERVs actively shape host evolution through complex mechanisms. This is not simple, involving innovation, genetic innovation. Yeah, evolution innovates. It's very teleological. Remarkably, certain ERV components perform critical cellular functions. These now constitute integral components of host genomic architecture and regulation. It is remarkable. But that's evolution for you. It is now well established that ERVs constitute integral components of all vertebrate genomes. The serendipity is off the charts. There's so many more examples of this. I had to pare back. I had way more slides than this. Another point I want to make and illustrate is the invoking of selection. So if you remember, the core assumption was junk. ERVs are junk, genomic junk. And we've just seen, well, no, they're not genomic junk. They're actually very interesting things. If you believe in evolution, they actually have incredible function. They are incredible, at least incredible function can be derived from ERVs. That means there's a fitness associated with ERVs, and that means that there can be selection. So now you're going to invoke selection. So the key premise of junk is no longer. And now there is function. And with function, there is location. What do I mean by that? Well, the function is dependent upon the location in the genome. And so now you have the location is under selection. So there's no longer junk, there's function, and with function there's location, and with location there is selection. That is going to be very important here. In other words, not only is the ERV integration site not random. Remember, we talked about that a few minutes ago. The first thing I started out with was this assumption that they're random. I said the integration site is not random. There's all kinds of factors that can influence the integration site. And I referred to that one paper, that one study, where they actually synthesized an ERV, and they watched it, and they noted, this is not randomly integrating. So that whole assumption of random integration was out the window. However, remember I said that after the integration, all kinds of things can happen because these are retrotransposons or transposable elements. They can move around. They can be deleted, copy and paste, and so they're mobile. Well, that whole process of post-integration dynamics can move these things around like crazy. And that can involve selection. Because as we're learning, oh, okay, there's function. And so there can be selection. And so there's selection bearing on this dynamic moving around process. That means they can be anywhere. And evolutionary theory can sustain any pattern because there was integration site and influences, and then there was dynamics after the integration by selection. So they can spin any story they want about how evolution did this, and they don't have to have any pattern. You can have a pattern that looks like common descent can be because it had selection involved. It needed to be in those locations. In other words, not only is the ERV integration site not random, but subsequent provirus location is subject to selection. And so it is highly non-random. This leads to a two-step integration fixation model. So there's the integration step, but when it integrates, it's only in one individual. And then you pass it down to you reproduce, it's in your children. That's not throughout the population. How does it get throughout the population? How does the entire population get this? It was only in one person. How does that happen? That's the fixation process where it becomes fixed. It takes over the population and becomes the majority, the vast majority of individuals in the population have that. Then it's fixed in the population. So that whole process of fixation is after the integration. So you have a two-step model. Integration and fixation, and both of those are highly, highly non-random. They occur according to a lot of different factors, and so the whole assumption of that video and of the argument is, oh, well, there's no reason. They fall into a common descent pattern. There's no reason. Well, of course there's reason. There's a reason for the integration site, and there's a reason for the fixation step. This two-step model involves all kinds of non-random factors. I discussed that one study that showed how some types of retroviruses prefer actively transcribed genes. That's hugely non-random.
Now, how much evidence is there that a virus can integrate and endogenize, so how can, is there evidence that we actually have viruses that have fixated and basically been able to endogenize like that?
Yeah, I mean, a lot of it is inferential, looking at things and inferring what happened. But I think it's also reasonable science, and probably does happen. So I think it's. I mean, these are all bigger questions, but I don't think this is going way out on a limb to say, yeah, it does happen. You do have integration and you do have fixation. I think that's quite reasonable. Yeah. So there's evidence that, the whole problem of how do these things get into the sex cells? That's a non-trivial question. Usually they're in in the throat and the lungs and so forth. But yeah, it can happen. So as strangely as that sounds, I think that's, would you say reasonable science?
Would you say even getting one, let's say, it does get into the sex cells and it's passed on to the next generation, but like you mentioned, it has to go through the entire population if we're talking about.
That sounds weird, doesn't it? How would I get an entire population? I know that sounds weird. It's actually good. It's actually good science. Yeah, that's reasonable.
Okay.
Now, it takes a long time. So
okay.
It's going to take many, many, many generations. So what happens is you get into one individual, and then a couple of generations later, it's in a dozen individuals, and it slowly proliferates through. If it's not harmful.
Okay.
So
you're saying there's no problem with that aspect of it?
Right. Yeah. Yeah, I am saying that. Yeah.
Okay.
Yeah, my position would be that there's no problem with that. Yeah, I don't think there's a problem with fixation of neutral or near neutral phenotypes. can propagate through a population for sure. Yeah. The importance of post-integration selection forces on the ERV provirus location is highlighted by ERV integration studies. What do I mean by that? These ERV integration studies reveal that observed ERV integration events contradict ERV location distributions in the genome. Okay, what do I mean by that? That study I talked about a while back where they actually they synthesized an ERV and they watched it integrate into cells, into the genomes, and they saw where it integrated, and it was non-random. Okay, they compared, it was a very ingenious study. They compared it to what we observed in the human genome. So they took human cells, injected this consensus retrovirus, and watched where it integrated. And they saw where it integrated in the genome. And then they looked at the actual distribution of this retrovirus in humans, in the human population. Where is it? So of all the chromosomes, and where does it land, and where is it in the chromosomes, and so forth. So they compared what they observe in humans to their laboratory experiment, and they weren't the same. So they saw that they did not integrate randomly; they were non-random. But then those non-random locations were not the same as what they observe in the human population. So that's where you say, well, in the process of fixation, these things moved around. There was influences on the location. So the two-step process makes sense. The two-step model makes sense. There's the integration step, which is non-random, but then there's something else happens as it becomes fixed in the population. That makes sense. So that's my point there, is that this is a great study. They compare observed integration events with fixed ERVs in the human population. These locations do not match. So my point is there's all kinds of moving parts here. There's all kinds of effects, causes and effects, both at the integration step and in the fixation step. So they synthesized an ERV and watched it integrate into the genome. Their synthesized ERV preferred genes and active transcription. Now, again, this is a huge step. Remember, they said in the video, and the argument in general, as you hear it from evolutionists, is there's 10 million or more of these sites across the genome, the human genome, and it just appears random. It's just random. It has to be. That is in the calculation. It's embedded in the calculation of that huge number. It has to be random. But here, they found in this study that in the integration step, when you just simply watch this ERV integrate into the human genome. Now, this is a different family of ERV, by the way, but it is a retrovirus. It's an endogenous retrovirus. It's an ERV-K. It preferred genes. The genes are a tiny, tiny fraction of the genome. And it preferred active transcription, genes that were actually being used, which is a tiny fraction of the genes. So no, it doesn't just, these things don't just, randomly integrate across these 10 million sites, which is the crucial assumption of that calculation. In contrast, these types of ERVs are, now, in contrast, you look at the actual human population, and they're found away from transcription regions. So there's the fixation process. As it becomes fixed in the population, things happen. So bottom line, a lot of non-randomness in the ERV location. The video's premise of random ERV location is false, just clearly from the science, false. Remember this slide here. For no reason. These things they observed them at the same locations in the genome for no reason. Wait a minute, where'd you get that from? What do you mean for no reason? Of course there's reasons. There's tons of reasons. That's a totally false assumption. Now, I'm not saying this is an open and shut case. I mean, this is an interesting question. There's a lot of uncertainty with these ERVs. There's a lot we don't know, but you can't just go and make these hard and fast black white claims and then these ridiculous calculations based on it. Therefore, the video's calculation is false. There's no question about that. It is a false calculation. You cannot trust that. Another interesting ERV provirus location bias is orientation. Now, what do I mean by that? So you got the double helix in DNA. You could be on one thread or another. So you can go in one direction or the other direction. That's called orientation. Human and mouse ERVs that are located in introns. Those introns are close to genes. They're in between gene segments. So they're right there with genes. Human and mouse ERVs that are located in introns, are mostly present in antisense orientation. That's fascinating. In other words, they prefer one of the two DNA coils. One of the, it's this double helix, right? One of the helices is preferred. So it's not randomly inserting in one helix or the other. It's preferring one of them. If you are in the region of a gene, well, that's a very non-random. It's an example of a very non-random pattern, but it's an interesting one. But ERVs found inside transcriptional units are strongly biased in the opposite transcriptional orientation relative to the host gene, thought to be less disruptive. Okay, but they observed no such orientation bias in their synthesized ERVs. So when they did the synthetic experiment in the laboratory, where they took the synthetic ERV and they put it into human cells and watched it integrate, there was no orientation bias. So the integration step didn't have that bias. But then when you look in the human population, you see, yeah, there can be that bias. So something is happening after the integration step and the fixation step. So again, just an intriguing, interesting example. This again reveals that ERV integration is insufficient to explain observed ERV locations. There's more to it. Need to consider both the ERV integration and the subsequent fixation process, the two-step model. Practically any pattern can be explained. And I discussed that way early in this video of how they have long since forfeited the claim that evolution requires a common descent pattern. Remember, I said when we looked at the actual evidence, the actual data from that study, there were X's. Certain species had X's, and the one particular ERV integration had 3 species that didn't have the ERV. And evolution is like, oh, well, there were deletions. They had deletions in that one. Oh, wait a minute, you're okay with deletions? Yeah, sure. These things move around, they delete. Any pattern is any pattern? Yeah, any pattern can be explained. Well, then how can you argue that one pattern forces us to believe evolution is true, common descent is true?
I've seen this too in papers and things talking about common ancestry, and it'll even say, like, if we didn't see this, it wouldn't be evidence against evolution, right? Right. So right. They basically say anything can happen. So this is a philosopher's dream. It is so much fun to analyze this theory from a philosophy of science perspective. It is all over the map. There are many explanatory mechanisms for where a ERV is found. Provirus deletions, provirus reinfections from exogenous retroviruses. So you can have an infection event that we talked about, these integration events, but then you can have another one. This can happen repeatedly. So you can have these retroviruses all over the place just because of reinfection. Or you can have transposition where they're moving around, copy and paste within the genome.
You can have genomic rearrangements. Remember, we saw the gibbon in chromosome 12, and that was still considered to be homologous because they just had a genomic rearrangement. Oh, okay. You can have purifying selection. You can have genome assembly errors, so the data aren't even right to begin with, et cetera, et cetera, et cetera. There are all kinds of explanatory mechanisms that evolutionists can call upon to practically any pattern can be explained. And the key premise of the video that ERV location in the genome is random is, again, falsified. These evidences indicate a two-step process for ERV location: initial integration and later fixation under selection. Anything can be explained. ERVs fixed in the human genome show a different distribution than has been seen for integration by any previously studied exogenous retrovirus. So in other words, this isn't just that ERV-K that they studied, that consensus ERV-K. This has also been seen in other exogenous retroviruses, which is, these are retroviruses, so it's meaningful. Therefore, the two-step model for ERV accumulation is likely operating on endogenous retroviruses. Here is a later study of.
Can you go back to that really quick? I didn't catch that last point.
I'm just kind of repeating myself here.
Okay.
That the two-step model, I went through it fast because
Okay,
it's just
yes.
hammering home this idea that the two-step model, integration and then later fixation for ERV accumulation, is likely operating on endogenous retroviruses. ERVs. ERVs are the ERV location that we observe is due to a lot of things. The initial integration preferences, and then later the fixation process. It's just a lot of moving parts, a lot of possible influences at work.
And that's only if we're assuming all these things that are called ERVs are actually ERVs, because they might not even be something that came into the genome.
I mean, that's a whole nother question. So I'm definitely kind of following the mainstream nominal understanding of all this. But you're right. There's when you have absolutely ridiculous, non-parsimonious conclusions that I haven't even gotten through all the problems, then yeah, you have to start looking elsewhere, and you have to open up your mind. So yeah, who knows what's going on here? Because you do get sacred cows in science over and over and over again, and they eventually topple. And it's like, oh, for centuries we were sure that this was the way it worked, and somebody fought that, and they lost their career because of it, and they were ridiculed, but they were right. Here is a later study of the two-step ERV location model. So this is a later study, 2016. Their model attempts to, quote, disentangle integration versus fixation preferences. So if you have these two-step, trying to just say, okay, good, they're trying to work on this. And they say, we do not completely understand, understatement of the century, we do not completely understand the fixation preferences. This is a hard problem. They're looking at this and it's like, yeah, this is hard. This isn't all making sense. So our knowledge of how the genomic landscape influences ERV integration and fixation is what? Still limited. Yes, definitely still limited. So far, what have we looked at? There are many ERV integrations. Remember I talked about in the human genome, there are hundreds of thousands of endogenous retroviruses, not 210. Okay. 210 was a filtered version. ERV location is not random. No. ERV is not useless junk. ERVs are not useless junk. ERVs and locations are under selection, a total breakdown of the video's premises, and additional problems such as serendipity, the ridiculous levels of serendipity required to explain the incredible functions that ERVs apparently have contributed. Now, next, how are you going to work? How is an evolutionist going to try and explain all this and kind of make sense of this? This is actually going to be a problem for evolutionists. So they're going to use teleology a lot. So teleological explanations are rampant in the literature. I'm not going to try and even cover this because it is so broad, but I'll give you a few examples. Teleological hypotheses, not just broad brushing things, but also deeply into the actual hypotheses. Teleology is a Trojan horse here. It's infecting like a virus. Teleological explanations are infecting the science. And so evolution is a deeply teleological theory, even though they reject teleology. There's this huge internal contradiction in evolutionary theory. I'll just give you a couple of examples here of some papers. This paper says a host. So what's the host? That's the person who's being infected, right? The organism that's being infected. A host, and remember, when a host is infected, that's a bad thing, right? That's a bad thing. To get infected by a virus is a bad thing. But as we're seeing, an ERV, a retrovirus, can potentially provide all kinds of good things. One of the good things that I didn't mention here is the possibility that the infection can then later be used as part of a defense mechanism against further infection. That's a more complicated process, but the possibility that the body's defense mechanisms can marshal and leverage infection sites in its defenses. So now you look at an ERV, at a retrovirus, at a viral infection, as maybe it's a little bit of a trade-off situation, right? There's good and there's bad, right? So they're going to use this explanation here. A host will what? Risk compromised genome integrity. That's an infection, a virus infection. Risk compromised genome integrity in exchange for what? Potential evolutionary catalysts. This is like a strategy. I've got my hat on. I got my calculator. I am strategizing.
And so like you kind of like think of it this way, where it's like, okay, the cell's like, yeah, this could harm me, but hey, it could give me that placental attachment I've been wanting.
Exactly. This is just dripping with teleology. These are like little strategizing agents. Continuing with this thought, thus for the host, quote, ERV acquisition. So it's acquisition. It's like an acquisition. The government there's an acquisition strategy of the government. They buy weapons from the defense industry. There's acquisition. Well, that's what cells do. They acquire things. ERV acquisition may be an effective defense strategy. See, there's strategies. Very teleological. These examples showcase the incredible resourcefulness of what? The genome. It's an entity. It has what resourcefulness is an attribute you assign to entities, not to just a piece of mass. Like, this cup here does not have resourcefulness, right? Resourcefulness is something you assign to entities.
A mind can be resourceful. A mind can strategize. A mind can innovate.
Right. A cup
but
cannot.
If you don't have a mind, you're not doing any of that stuff.
Exactly. The internal contradictions in this theory are just hilarious. ERVs are what? Agents of genetic innovation.
Again, if you don't have a mind, you're not an agent.
That's all I'm going to say about the teleology, because I could we could spend all day on this. It's so rampant in the literature, but I wanted to show at least a few examples of this is what happens when you do this. Okay. Next, I talked about early on, how there's this the virus structure as the genome in the middle that codes for proteins, and these flanking sections. And when you integrate into the genome, you still have that genome section, that proviral body, as they say. But the caps now have a different name. They're called LTRs, lateral long terminal repeats. Those are different. They're a processed version. They contain a lot of regulatory information, but they're a processed version of the virus particle that's floating around. It has those caps, those end caps. Those are processed. So you end up with something we call LTRs. And I said I'd come back to that. This is very important. How did that come about? How did viruses evolve? There's got a lot of proteins in there, and then you got these end caps, and these end caps get processed, and this processing is very important. Well, this processing step, we call that integration. It's part of the integration step. It gets integrated. What happens there? Well, in the virus, in the retrovirus, you don't have DNA. I haven't gotten to that detail yet. I've just been calling it the genome that codes for proteins. Well, there is no DNA. There are DNA viruses. These are not DNA viruses. These are RNA viruses. It is RNA in there, slightly different, but still containing nucleotides, coding for proteins. It's a single strand. It's not a double strand. So it's a single strand RNA. That is going to be, there's going to be a complementary strand copied, and it's going to be converted into DNA. So you're going to end up, as part of this integration step, you're going to have a double helix DNA that gets injected into our genome. And you're going to have these LTR end caps synthesized. That whole process is amazingly complex. And there are molecular machines that do that. Where do these molecular machines come from? Well, they're part of the genome. So it's carrying this virus particle is carrying everything it needs. It's got genes in there that will do the processing, but those genes need to those genes need to be converted into need to be used to create some proteins. Those proteins then will do the work of doing all this processing and injecting it into the genome. Don't worry about all the details here. My point is simply, this is a very elaborate, complex process. This is non-trivial. It doesn't just, it's not natural. It's not just like, oh, it just slips into the genome. No, there's a lot that happens here. And there's no plausible explanation for this under evolutionary theory. In other words, there's no plausible cause for the evolution of retroviruses. There's no such thing. If these viruses are such powerful evidences for evolution, as they're claiming, don't you think they would discuss the evolution of viruses? Let me repeat that. If viruses are such powerful evidences for evolution, then aren't you going to tell me how they evolved? I mean, you must have a story of how they evolved. That must be part of the evidence, right? That, oh, we know how they evolved. No, we don't even know how they evolved. How can you claim there are powerful evidences for evolution? In other words, you're resting on this an enormous contradiction. You're resting on a contradiction of your theory. You are relying on something that is contradictory. You're relying on the evolution of retroviruses. How can you even explain
Now, they're
that?
going to say to that, though, they're going to say, well, just because we don't know how it happened doesn't mean that, it didn't happen. So we can't know. It doesn't mean it's a contradiction. So what do you say to that?
You're just making an argument from credulity. Oh, you just don't know. You just can't imagine. No, it's much worse than an argument from incredulity. It is our negative evidences. What we know from science is that they could not have evolved. Now, that could change tomorrow. Wonderful. Anything can happen. I'm talking about today's science. I'm talking about what we know from science. It is a contraindication. Retrovirus evolution is contraindicated.
In what ways? Do you mind talking about that a little bit?
Well, it requires a whole bunch of proteins. This genome has all these genes that code for proteins. The evolution of a single gene is a problem. That is an enormous problem. How is evolution going to evolve these genes? One of the key genes in there is the gene that is the protein machine that actually does this, turning the RNA into a DNA and then injecting that into the genome. It's called reverse transcriptase. And evolutionists used to believe that evolution flowed from the DNA to the body. It's a one-way street. You have information in the DNA, and that then creates proteins and RNA gene products. So it's a one-way street. Reverse transcriptase contradicted all that. It shows that no, it can take information and put it back into the DNA. It's way more complicated than evolutionists ever envisioned. Well, you have one of those, a reverse transcriptase that is tailored, and it works in a tailored way for retroviruses, and that's coded for in the genome of the retrovirus. So it's got the code in that RNA genome that's used to create this reverse transcriptase machine that puts the viral genome into our DNA. There's no explanation for how these genomes, these immense genes, could have evolved from random mutations. But then when you look at the actual integration process, it is immensely complex and elaborate. The reverse transcriptase and associated machinery are doing all kinds of things to get this to work, to process the LTRs. And this is crucial. You have to have these LTRs. There's no evidence of like intermediate steps, or there's no evidence how this could. I mean, this is so outrageous. I have trouble even communicating it. You have to have one mutation at a time to build this up, and then you have to at each step it has to be a functioning something to be selected for. It's just we're not even close.
So now, you're saying basically, if you want to be scientific about your approach, right now, the most scientific idea is that this cannot happen by natural processes. And that may change tomorrow.
I don't say cannot happen, because that's a black or white statement that you want to avoid in science. It's contraindicated. It's not likely. It's unlikely. It's there's the Eugene Koonin paper where he tried to estimate the probability of a simple entity that reproduces itself, and he came up with 10 to the minus 1,000. It's not impossible. It's astronomically unlikely. And so you get these, it's just ridiculously unlikely. It's not in science. We try and come up with likely explanations and things that they do a good job of explaining nature. How does nature work? And so, it's just what science tells us over and over and over again are these things are unlikely to have evolved by random mutation and natural selection. It's just unlikely. It's not a likely process. That's what science is telling us, the overwhelming preponderance of the scientific information. Now, this whole integration process is an example of that. And so how could that have evolved? Vincent Racaniello is a researcher, professor, who this is his specialty. And he's done a lot of really good lectures, and he has a lot of these lectures on the internet, and you can see them on YouTube and so forth. And here's one in particular. I'm posting here the URL, and just watch the video and you realize the incredible complexity and elaborate process of the integration step, and what these molecular machines are doing to create the LTRs, these end caps, and injecting it into the DNA. This is not just, it just happens. No, it's a very elaborate process that these molecular machines are going through. This all had to have evolved somehow. There's no explanation for that.
And we're just talking about one little thing that happens, and you've got so many things that have to happen in a single cell.
Exactly.
Right? So
exactly. Yeah. So it's just a good resource there to kind of get, it's easy to talk about these things in the abstract and to, just push these numbers around and come up with these calculations like they do in the video. When you look at something like this, how are you claiming this as evidence for evolution? Okay.
Mm-hmm.
We have yet another problem. So there is no plausible cause for the evolution of viruses. That they don't even talk about that. Yeah. But that's a huge problem. Now, the last thing I wanted to say, there's one more thing I wanted to say. And then we touched on this earlier, was the video and this argument in general is not an argument for the plausibility of common descent or the compellingness of common descent or evolution. It's an argument against the fixed species view. That's what the whole, that clip that you played there, remember, he said, any rational person needs to set aside the fixed species view. That's interesting. You notice he didn't say common descent or evolution. He said the fixed species view. What was the subject? What was the topic? What was the subject? The video says any, quote, rational person should, quote, set aside the fixed species view. The subject is what? The fixed species view. That's the subject. Nothing about these ERVs actually helps evolutionary theory. It is an argument against a different theory. This is an argument against a different theory. That's what this is. That number that he showed, it is a proof by the process of elimination. If you have theories A and B and you can disprove B, then only A is left. Theory B is what? Creationism, including the fixity of species concept, which goes back to Linnaeus in the 18th century and before. That's what they called it, the fixity of species. What is being evaluated and tested here is not evolution. It is the fixity of species theory. Evolution is declared to be true by the failure of the fixity of species. Now, you can only do that if that's the only alternative. So you've got evolution and you've got fixity of species. That's it. If the fixity of species is false, then evolution is true. So if you have a space of all possible explanations, every possible explanation for the origin of species, there's only two. So you cut it in half, there's two. Evolution, in various forms, of course, there's different variations on that theory. But in general, there's evolution and fixity of species. That is a religious argument. It's religious for several reasons, actually. One reason is you are claiming that the fixity of species model is the way that creationism works. That's a religious argument. But you're also claiming those are the only two possibilities. And so when you have that, in statistics and mathematics, we call that complementary. They exhaust the space. There's only two possibilities. One must be true and one must be false. They are complementary. They are mutually exclusive. There's no overlap and there's no gaps. There's no other possibilities. How can you know that? How can you know that all possible explanations for the origin of species are only one, there's only two of those? There are no other possibilities. How can as a scientist, you know that it's impossible. That's not within science. That's not within the purview of science. That's metaphysics, or what we say, religion or theology. That's not science. Scientists don't talk that way. You can't make those sorts of arguments in science. This isn't about science. This never was about science. That's why evolution is not a scientific theory. It is a religious theory. This is all religious with a lot of science window dressing. And so that's, you know, it's easy to get lost in the science and all these things I've been talking about, all the evidences and all this stuff. And that's interesting. That's important. This is all religion. And that was evident in that clip that you showed, although most listeners will not catch that. But in that clip, he says, you know, look at this huge number. Any rational person should set aside the what? Fixed species view. Ah, that's religion. That's religion, because that's what you are claiming is creationism. And you're claiming that it's mutually exclusive. It is complementary to evolution. That you cannot know that as a scientist. It's not, I'm not making a minor point. I'm not making a nitpicky point. This isn't like philosophers arguing. This is huge. It is a very important point to understand.
Wow. Okay. So I'm sorry.
So we have bad science driven by what? Religion. We said that I showed all the bad science. Like, this is just ridiculous. Driven by this. So that's it. That's the explanation of that video. There's a lot more to say, but you know, that gets the main points across.
Okay, good. Well, we had some questions and comments that I'll put up, but that kind of brings me to what I really wanted to ask you, because I've been hearing like evolution is the scientific theory, right? And I've kind of probed asking some people like, you know, is there any other possibility, like, you know, for a scientific theory? And there really isn't. So that causes me to say, well, does that itself expose it as a non-scientific theory? Meaning, like, if we can't even think of another possibility, I mean, what do you think about that? Am I making too much of that, or is it a strange thing that it's the only scientific theory?
Yeah, so this is another really interesting area for philosophy of science. Does science explain everything? Now, I'm not anticipating a single answer to that. There's no right answer to that. But you have to decide, is your science that you are going to pursue complete or incomplete? Do you have completeness or not completeness? So completeness means it explains everything. And so if it explains everything, that's a huge, you know, burden for you to carry. Now, another question, set that aside for a minute. Another question is science restricted to naturalism? And now there's a lot of people today who will say, well, of course, I mean, that's by definition, that's what science is. No, that isn't. That's not that's not by definition. That is a claim. And again, there's no right answer. There's no one right answer to it. It's not it is or it isn't. That's your science. So different people can pursue different kinds of sciences, but you can say, I believe, I insist, I'm going to make sure that science is strictly is restricted to naturalism. That's a form of science. In the 17th century, at the Royal Society, there were a lot of people who argued, no, it's not. It's not restricted to naturalism. I want to be able to pursue anything. So there's different positions on that. So you can have completeness or not. You can have naturalism or not. You can't have both. What if you say, I want to have completeness and I want to have naturalism? So I'm saying I can explain anything. And when I say anything, I'm including, you know, love, hate, emotions, the mind, human experience, the galaxies up in the sky, I mean, everything. That's everything. Everything that we know of, it can be explained by science. And science is restricted to naturalism. So you have both. Well, you have all, that means at the very beginning, before you even started, you are making the assumption that everything is explainable by naturalism. That's called metaphysics. That's before the science begins. You've already restricted it. You put science in a box. You have no scientific reason for making that claim. What experiment did you perform that informed you that you could explain everything by science, and you could, and then it was all naturalistic? So everything, all phenomena are explainable by naturalism. How do you know that? You don't. That's your position. Fine. If you want that position, you can have that, but you have to understand that's not science that is informing your position. That is a metaphysical position. That's your starting point. You're informed by some other considerations, like religion. That's always the other considerations, is religion. So you are religiously driven to do that, and that's fine. Go and do your science that way, but it needs to be understood. You can't just brush that under the rug then and then start making truth claims. You have found that evolution is true by virtue of your starting point, and it's not good science. It's terrible science. It's ridiculous. So you can't then be claiming, oh, well, it's true. We've discovered that evolution is true. So that's the kind of thinking I've written on this quite a bit more than that, but that's the kind of thinking you get into, right? When you start asking these questions, well, what does science do? And you get heavily into the philosophy of science, and that's important. It's not just academics arguing in the corner. This is really important.
Yeah. Okay. Thank you so much for answering that. And there's some comments and questions. So Frank says, excellent presentation.
Oh, thank you.
And says, okay, are there known ERVs that are in only some members of a particular species, but not all members of that species?
Yeah, that's a good question. So I do believe there certainly are. The answer is yes. There are. You can get. Let me just say that's the fixation process, the process of becoming fixed. So it's in some individuals, but not the entire or the majority of the population. When it reaches the majority or near majority, or the majority of the population, it is said to have endogenized or become fixed. So it is then. Oh no, I'm sorry. Let me make it clear. It is said to be endogenized when it goes through the sex cells and is then being transmitted to all of the progeny. But when it becomes the majority of the population, it's fixed in the population. So yeah, there are. I do believe there are ERVs that have gotten through the sex cells. So it is this. So the E it has attained the endogenous status, but has not yet become fixed in the population. It has not spread throughout the entire population. I do believe there are examples of those. I can't bring them to mind. This is not exactly, I can't bring those to mind, but I do believe there are some examples of those. So it's certainly, let me just say, it would be something you would expect. I would expect that. It wouldn't be a surprise. It's something I would expect.
Okay. So Hamobu says he doesn't understand probability. He thinks that unlikely things don't happen, but they happen all the time. It's unlikely that any one person will win the lottery, but it's certain that someone will.
Yeah. So this is a probability explanation. You can turn that around on the video itself and say that they don't understand probability. So remember what the video said. There's a retrovirus that is found in a particular location. in one species. So the human has this retrovirus. You look at the chimpanzee, it has the same type of retrovirus in the same location. You then look at the gorilla, and it has the same retrovirus in the same location, right? We went through this. And what did they say in the video? That's unlikely. That's super unlikely. And they had that number, the one with all the zeros. It's one chance in a gazillion, 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 right? And so Hamobu, what would you say to that? You would say, well, they don't understand probability. They think that things are unlikely because but those things happen all the time. So your criticism applies to the video. They are saying that. Now, the fact is, it does, the logic in the video is valid. It's a valid piece of logic. It's a valid piece of logic to say that it is unlikely to have happened. So this argument is not, it does not apply to what I'm saying. I'm saying that the science, it does not, I've written all about this, but it's hard to explain off the top of my head. But let me just say that your logic applies to the video, and I hope that's obvious.
Okay, next question. R. Brown says, if humans didn't evolve from lower life forms, then can you explain why a human fetus temporarily has a cloaca, which is a kind of genitalia typically found in reptiles and birds?
Yeah, so there's all kinds of embryonic development pathway arguments for evolution. You look at the embryo and it looks like other species. Now, having not created the world and having not created the human species, I can't explain everything. So when you ask that question, well, how am I supposed to explain that? But then again, that's not just me, that's everyone, right? Evolutionists can't explain that either. What this argument boils down to is, if there's no reason for this there's no functional reason for that. And we just don't know that. We don't know that there's no functional reason. And
would you say this is one of the religious arguments that you're talking about? Like, God wouldn't have done it that way.
Right.
Therefore, it's evidence for evolution.
That's what it boils down to. That's what the strength of the argument boils down to.
Okay, so next one from Hamobu. Why would God need complex cellular machinery that makes non-living matter into living beings? Why doesn't he just make living beings?
By fiat, by miracle, by just speaking it into action. So you could argue, well, he did that. So isn't that what it says in Genesis, that he did that? But I think that doesn't answer your question. I think your question is, why doesn't he do that all the time? Why don't we just have miracle? I want to have a child, so just boom, you have a child. And so this is a religious question, a religious argument. Why doesn't God do it this way? Why doesn't God do it that way? Not being God, I don't know, and I don't. I didn't. And I'm not. I don't hold. It doesn't make any difference to me whether God wants to do it this way or that way. So to me, it's not a big deal. Like, well, why didn't God do it that way? I don't know. God does it one way or another. That's the way he does it. What is important to me is when religious ideas are paraded as science. So you have a religious idea, and then you say, well, this is scientific truth. So we need to be real clear. Yeah, we can have these religious discussions. Why didn't God do it this way? Why doesn't God do it that way? Well, I don't know. I'm not God, so I'm not a very good source for that.
Yes. So Mr. Green says religion and science aren't at odds. Thank you, Mr. Green. Travis wants to know, does creationism have any falsifiable concepts that can be tested?
Well, you'd have to ask creationists. I've heard certain creationists make those sorts of claims. So that there probably are. There's a lot of people who have said evolution doesn't have any falsifiable concepts, that everything can be twisted around. Anything can be explained under evolution. So, there's always these arguments and there's always answers. I do think that there are certain evidences that can falsify evolution, and I do think there are certain evidences that probably could falsify creationism. But when you actually have those evidences against evolution, they will deny them, or they will say, well, we'll figure that out. For example, there's just all kinds of evidences that falsify evolution, that they just say, well, we're working on that. So, these are more philosophical questions that, all sides, all theories have to confront.
And so you said, like basically you have to ask a creationist or something like that. Like, do you consider yourself, how do you consider yourself with this? Do you make claims about creation?
Yeah, so I don't consider myself to be a creationist because I don't agree with their claims, but I'm more interested in what the science tells us. I go by the science. And when I see science being abused, then that, I get interested in that. I'm a Christian, and I believe that God created the world. And I'm not exactly sure how he did that. Right? It doesn't bother me if God used secondary causes or primary causes. In other words, miracles or natural law. It's like, okay, fine. It's not a big deal to me. My day doesn't get ruined if God did it one way or the other. I'm not big on that. Things that I can't nail down from the Bible, I don't get excited about. So if the Bible tells it to me real clearly, then yeah, I got that. But if there's ambiguity, I'm not quite sure for the Bible, then I'm not going to pour something into that meaning. Now, back to this creationism thing about falsifiability of creationism. It is what I think what's important to understand is we gave it our best shot with evolution. Man had religious requirements to get God away from the world. And so we needed to have evolutionary theory to explain the species. That was real important. So ever since the Epicureans, we've been working on this theory of how the world arose naturalistically. And the scientific evidence is abundantly clear that idea fails. So when you say, well, is there anything falsifiable about creationism? I mean, I'm not sure what form of creationism you're talking about. I don't know what you mean, but the point is, that's really kind of grasping at straws. I mean, the science is so abundantly clear that evolution has failed. Why are you worried about whether creationism has falsifiable concepts? It probably does. I mean, if I was to guess, I would say if evolution worked, that would be a falsification of creationism. So I kind of agree with I'm kind of in line with the usual argument that, yeah, if the evidence is powerful and compelling, and if evolution was a fact like they say it is, then yeah, that would be a pretty good falsification of creationism. There are creationists who say that God can do it fully naturalistically, 100% secondary causes, and he can use evolution. Okay, so they would not be bothered by evolution being proven true. But I think that that would be pretty good falsification of creationism. But we're nowhere close to that. I mean, we're at the opposite side of that. The science is so problematic, and it's not really surprising, right? I mean, you look around the world, look in the mirror, look outside the window. Do you think that this all arose by chance? That's kind of a heroic theory. Well, it's failed. It's failed by your intuition, but it's also failed by today's science. The complicated science of biology and physics has falsified this over and over and over again. So I think the number one way that you would falsify creationism, to answer your question, is by the success of evolution, and that's just failed so badly that it just seems like your question is really kind of grasping. You're kind of like trying to avoid the obvious, like, oh yeah, well, evolution is false, is all problematic, but can you falsify creationism? Well, why are you worried about that? I mean, the science is so abundantly clear that evolution has failed. There has to be some other explanation. What that is exactly is a complicated question, a complicated problem. But I wouldn't. My first question wouldn't be, well, can you falsify creationism? It's more so, gee, this is interesting. Our best shot at explaining the world naturalistically has fallen on its face over and over and over again. What else might we have here? That should inform your worldview, should inform your view that we need to think more broad. Maybe this whole naturalistic project was a bad start to begin with, and that it came from false theology. It was motivated religiously, and it forced us to investigate this brand of science, this strictly naturalistic explanation for origins, and it failed. So that should tell us something, that those religious ideas weren't right. What were those religious ideas, by the way? What was it that got us going down this path? Why did we do this? Oh yeah, that's right. It was to distance God from the world. God must be distanced from the world. And why was that? Well, there was a bunch of different kind of deism sorts of ideas that people are infatuated with. That we have to distance God from the world. God cannot be immanent. So immanent is an important, theological doctrine that God is in the world. God is active. He's in the world. He's here. He's not on a distant Mount Olympus meditating while the world rumbles along. That's the evolutionary project. Get God distant from the world. Well, that failed. So there was a religious premise, a religious belief, we need to get God distanced. That led us down this path of Epicureanism and evolution. That failed. So therefore, by modus tollens, the original assumption fails. If A implies B, and if B is false, then A is false. So the original theological premises that God must be distanced, we got to get God away from the world, that failed. So maybe we should consider the Bible instead. Now, I'm not talking about any version of creationism. I'm not talking about Genesis. I'm not talking about 6 days or anything like that. I have no idea about that. I'm talking more generally, because that's what's at stake here. Forget about don't get wrapped up in the who, when and where and how did God do this or that. Was it 6 days or the age of the earth or the mechanisms, the miracles? That's all superficial icing on the cake, so to speak. What's more important is the underlying theological motivation for evolution, which was to get God distanced from the world. That was crucial. I've written a lot about this. It's not just one religious belief. There's a dozen or so theological traditions that insist that God must be distanced from the world. And that led us to evolution. Evolution failed. So those underlying theologies, much more important, I think, than the details of Genesis 1. I think it's more important, these underlying doctrines of God and how God interacts with the world. That's what drove us to evolution. Evolution is false. So those doctrines, which are non-biblical, by the way, maybe we should consider the Bible instead, and how God is immanent. God is in the world. He's here. He's now. The kingdom of heaven is here. It's among you, as Jesus said. What was the phrase? Jesus. There was no distancing when Jesus speaks. Read the scriptures and listen to Jesus. it's all, here and now. It's a whole different kind of proposition that he's laying out than the evolutionary view that God has to be distanced. So to me, I'm trying to get people to focus on those things, not on, creationism. This, don't be twerked by, the Kentucky, San Diego, Australian creationism stuff. Forget about that. It's not relevant, in my opinion. It's much more important the core theological drivers that are driving evolution and the failures of those, and the biblical theologies that are true, that actually are consistent with science. They don't lead you to bad science. They lead you to science that makes sense. To me, that's way more important. So I know that's a long-winded answer, but hopefully that helps fill in that there aren't simple, one-liner answers to, creationism is true and evolution is false and all that. Just get away from this tribal stuff and get into the scripture and actually what it's actually saying. And I allow for, there's parts of scripture that are poetic, right? And there's parts that are symbolic and metaphorical. Maybe Genesis 1 is, I don't know how to interpret that exactly. That's a hard question. So I don't mean to be punting on Genesis 1 altogether and saying, don't pay attention to it. All scripture is important, but it's, I don't think we should get hung up on where scripture is difficult to interpret. Don't claim that you be. I guess I'm comfortable with having some uncertainty about it.
Yes, and I have to
sense at all.
Yes, absolutely. I love it. It's perfect. And Mavis says Rebecca eats up everything Hunter says and rejects actual evidence. The incredulity runs deep here. Okay, I have to respond to this because I want to just say, yes, I do eat up everything Dr. Hunter says. And that's because in him, I have found a person who knows the science, the philosophy, and the history of all of this. So he has his methodology for me is perfect because he's just trying to figure out what's true. And I feel like he approaches it in a very non-biased way. And he didn't have the starting, presupposition that, hey, the Bible is true, this one way, basically one way of understanding creation. Even though I'm a young earth creationist, I don't like that as a starting point, because how can you figure out if you should believe the Bible? So you have to be able to have a bigger, a broader way of trying to figure out what is true. So I just find Dr. Hunter to be very perfect in the way that he goes about things and has such a broad knowledge. So yes, I eat up everything he says. Thank you so much. But I don't think it's about incredulity. Okay, so Casper Holmes says, and I know you've already addressed this in previous things, but he says all living things are genetically connected through common ancestry. DNA is evidence for evolution. I know we had a whole show about this, and Casper, I would recommend that you go back and look at that. But do you want to address it quickly?
Well, just quickly, he's saying DNA, not the DNA code. So he's saying DNA. So there's a difference. So the DNA code is the way that the information in the DNA is coded for proteins. And we had a show on how that's claimed to be evidence for evolution, claimed to be a slam dunk proof of evolution, but it's actually all false. And it's a problem for evolution. But what Casper is saying here is DNA is evidence, the DNA itself. So he's referring to the structure itself, the double helix structure. That's evidence. Now, that can be considered to be evidence for evolution in a couple of different ways. One way is just the the fact that it's a homologous through all structure, all species. So all species have DNA, right? So it's like, well, that's unbelievable. You know, so it has to be common descent. I mean, why would all species have this DNA? And so that's an argument for evolution. That's a powerful argument for evolution, but it's religious. It is a religious argument because it comes from the principle of plenitude. You can see my videos on principle of plenitude. It is an argument that God, if he created the world, would have used more diversity. He would not use the same design over and over again. From a strictly scientific perspective, DNA is an enormous problem for evolution on a number of levels, but you can't make an argument why DNA is evidence for evolution from a scientific perspective. So yes, you're making an argument here and you're making a strong argument for evolution, but it's religious, it's theological. And that's the incredible subtlety and the deception that's at work here, is that I'm sure people like Casper, and I'll speak more generally because I'm not sure exactly what Casper was getting at, but people more generally will make these sorts of arguments, asserting them as though they are science, and they're completely convinced they're making a scientific argument, and they're not. And it's the deception is very powerful.
Okay. Hama says the pattern of distribution of ERVs among different species exactly mirrors the pattern of similarity between all other genetic markers. It elucidates the nested hierarchy evolution predicts.
Right. That is exactly the false premise of the video and of this argument. That is the basis of this argument. That endogenous retroviruses fall into a common descent pattern. No, that's false. They don't. So you're making an assertion that doesn't come from science, and so it is a confirmation testing. So you look at endogenous retroviruses that fit your pattern and you hold those up as your evidence. There are hundreds of thousands of endogenous retroviruses that do not exactly mirror the pattern of similarity between all genetic markers. They don't fit. They're one-offs. And under evolutionary theory, they are said to have inserted recently, so they're not in other species. So, you have to come up with more. You have to come up with the details of the evidence that you're pointing to, because they just don't. That is a misrepresentation of the scientific evidence. It is true. So I think what you're saying is that there are herbs that do fit that pattern. Yes, I agree, there are. But as we just talked about in this video, there's all kinds of problems with that, and including the fact that there are others. There are a lot that don't. There are a lot of herbs that don't fit that pattern. And then you've got the serendipity, and you've got the fact that there are, for those herbs that don't fit that pattern, evolution has no problem with that. They can explain those perfectly well. So evolution doesn't make a prediction here. Evolution is perfectly fine with common descent patterns and non-common descent patterns. So if they're the one no particular outcome can confirm the theory, if the theory is predicting all those outcomes, all the things we talked about. I won't rehearse them all here, but.
Okay, Casper says she believes the Earth is 6,000 years old. Really? Okay, maybe a little older, but don't get hung up on that. Like Dr. Hunter said, I am not hung up on this idea, one particular idea of creation. And there's lots of different things, but I think what Dr. Hunter said is what's important, is that when we look at the world, there's good reason to believe that this is not the product of natural processes. And so that's what my purpose of my channel is. It's not to convince you of a certain view of creation, but to help you see that God is real. God loves you. He's given you evidence for his existence in what we see around us. And that's what I want people to see. And I think people have been in a bit of a mind fog because of evolutionary ideas that have kind of blocked people from being able to see the truth about reality. Did you want to say something about that, Dr. Hunter?
No, that's a good point.
Okay. Charles Oliver says, how did you determine what method was used by your deity? Why do you equate evolution to religion?
Yeah, so the first question is I didn't. So I can set that aside. For the second question, why do you equate evolution to religion? Because evolution makes claims about God. So in my book, when you make claims about God or the attributes of God, that's religion. And last time I checked, that's not science. So it's all through the evolution literature. In Charles Darwin's book on the origin of species, chapter 6, he states that he is a theological utilitarian. And his theory is theological utilitarianism. It entails theological utilitarianism. Chapter 6. Now, there's religious arguments all through the book, but I like that one spot because he comes out with it, that theological utilitarianism is entailed by his theory, and he clearly is a theological utilitarian. That's a religious belief. That's a claim about God. There's about a dozen different claims about God in the evolutionary thought, in the history of evolutionary thought. And these run up to today. I'm not talking about, dead white men from hundreds of years ago. This is today. In today's literature, these ideas persist. So when you make claims about the attributes of God, you are dealing with religion, not science. And you can go to my channel and see I've got a playlist on theological naturalism that goes through all this. I've got another playlist on why evolution is a religion, why it's a religious theory. I've got journal papers, peer-reviewed journal papers on this. You just need to read the literature, look at the history of literature and the current literature. Ask yourself, why is evolution a fact? Now, the reason I'm bringing that up is because that's the consensus claim in evolutionary thought. The consensus claim is that evolution is a fact. There's different terminology for that. Different words are used. Some people say true. Some people say beyond all reasonable doubt, or whatever words you want to say. But why is it a fact? And there's several different arguments. The evolutionists are not shy about making these arguments. So there's a lot of books that have been written, Jerry Cohen's book, and a lot of others going back to Darwin. I've written about these in my books, and I've examined them. So if you look at all the different arguments that have been made since Darwin, you can collect those. And I love that. I'm a collector of arguments. And all of those arguments that prove evolution to be a fact, and they are valid arguments, they rely on theological premises. I'll give you one example. There's a 2016 paper, lead author is Baum, B-A-U-M, out of the University of Wisconsin. And it looks at the wealth of data, most of it's genetic data, but they also look at morphological data that have been accrued over the decades. So they're looking at a ton of data, and they are claiming that these data prove evolution to be true. Well, how do they do that? It's by virtue that they're not random. So the genetic data, the fossil data, morphological data are not random. When you look at them across the different species, they have patterns. They have non-random patterns. So similarities between the chimp and the human and all this. So many of these arguments go back to that there's these patterns, non-random patterns. Now, why does that prove evolution to be true? Isn't that a strange conclusion to make? Well, the idea is that if there are non-random patterns, evolution must be true, because God would not have created a world that way. When God created the world, he would have used all the diversity that he could have used. It would have been as full as possible. You wouldn't have these patterns, unnecessary patterns. That's a premise. You can make that premise if that's what you want to say, but it's a religious premise. And so that goes back centuries. That there's a history of thought there. So when you have this bomb paper from 2016, it's not just coming out of the blue. It exists within a tradition. These are metaphysical, theological traditions. So people read these papers and they go, "Oh, evolution is true," not understanding the history of thought that's behind it. There's a long history of thought insisting that God, and it's called the principle of plenitude and the principle of sufficient reason. Those two go together, that God would not create these unnecessary patterns in the world. And so here you have a modern peer-reviewed paper proving evolution to be true, all based on this principle of plenitude and principle of sufficient reason, which goes back centuries, and theologians have argued about that for centuries. It's a theological argument. So why do you equate evolution to religion? Because it's all through the literature. It's all through the literature. When you prove evolution to be true, you are proving it to be true on the basis of religious premises about God. And that's why I equate it with a religion.
Okay, Hannah says, I challenge Dr. Hunter to present any peer-reviewed research paper that falsifies evolution. Now, I'm glad that you brought this up, because that gives me a chance to share in the description of this video. You will have a link to Dr. Hunter's, I think there's like 22 falsifications of evolution, and he's got lots of research papers in there, peer-reviewed research papers that show how the contradictions in evolution. So please do go look at that. And as he mentioned, he has a channel called Darwin's God, and there's so much good stuff on that channel. And in fact, that's how I found Dr. Hunter, is just I found his channel when I was looking up something. And there's so much on there that is really excellent. If you guys have ever heard this idea that, like, oh, the human eye is basically not, it's not a good design, and the octopus eye is better. He has an excellent video on that, and two videos on that, and it'll blow your mind, and many other things. So just wanted to recommend his channel. It's called Darwin's God, and the link is in the description of this video.
Let me say another word about, thank you very much, Rebecca.
Mm-hmm.
Can you bring that question back up?
Oh, yes.
Because I want to say, so thank you again, Hama, for your points and questions. And what you're asking here is, I challenge you to present any peer-reviewed research paper that falsifies evolution. So as Rebecca pointed out, I have a paper on the false predictions of evolution. And these are fundamental predictions. And like Rebecca said, that'll be in the description below here on this video. So these are fundamental predictions of evolution. These aren't tangential. And there's a couple dozen in there almost. And they are all false. And you can look at the peer-reviewed papers for each one of those. You can look at the peer-reviewed papers that establish it as a prediction and establish the falsification of it. So that's an important finding, that evolution, all of its major fundamental predictions have turned out to be false. That's important, right? Now you say falsifies evolution. So the question becomes, does that falsify evolution? If you have a long trail of fundamental predictions that turned out to be false, does that falsify your theory? Well, there's no one answer to that. There's no right answer to that. You get into the difficulties of the philosophy of science. So in that paper that I have there, the false predictions, I talk about, does that falsify evolution or not? And what does it exactly mean to have false predictions? So this gets to a deeper question. Is there actually an answer to your question? In other words, is evolution falsifiable? No other scientific theory ever could have sustained this level of failure and still be considered a viable working hypothesis, or something we should even talk about, let alone a fact, a true theory. It's claimed to be true, even though it has failed over and over and over again. And so I would challenge you to actually come up with a falsification criteria. How could you falsify evolution? It seems like it's so elusive. How could you have the degree of failure? There's no plausible cause. How did evolution create the world? How did the entire biological world was created by evolution? How did that happen? There's no plausible cause, and yet it's considered to be a fact. It's true.
Now, he says, Dr. Hunter, you are the person claiming that evolution has been falsified, and yet you dance around without citing any specific study or data that indicates this is true. I'm telling you, he's got, you can go and look. He's not dancing around it. In fact, we plan on every week or twice a month showing you all of the falsified predictions of evolution. So we are going to get specific, and we have been getting specific if you look back at the times that Dr. Hunter has been on the channel, and you can read that paper.
Let me say something to this question. So Hama, listen very carefully. I did not claim evolution has been falsified. I did not say that. If I did, I misspoke. Maybe I said it, I may have said it, but I would have misspoke. I don't think evolution is falsifiable. So I would never say that evolution has been falsified, because saying that evolution has been falsified is like saying that Hinduism has been falsified. You can't falsify Hinduism. You can't falsify a religion. Evolution is a religious belief. It's a religious idea, because religion runs all through it. All of the proofs of evolution rely on religious premises. So the science is window dressing. Now, I did say that the science contradicts the theory over and over again. So you have false predictions. You have the failure to have a plausible cause. There's no plausible cause for the origin of species. There's arguments between evolutionists, scathing arguments between evolutionists, because they can't because the theory fails so badly. They have all these different hypotheses, because there's no one good hypothesis. They're coming up with new ones as we speak. Today, there's new ideas coming out. These are not minor tangential ideas. These are major reworks of the theory, rejections of the modern synthesis, because it didn't work. So you have major problems in the theory. Is it falsifiable? No, I don't think it's falsifiable, because is motivated by religious ideas. So that's point number one, is you are the person claiming that evolution has been falsified. No, it's contraindicated. It's contradicted by the science. I don't think it's been falsified. And yet you dance around without citing any specific study or data. So that's not true. No, actually, I have cited specific studies and data. So for instance, in this video that we're finishing up here this morning, I have a lot of papers in the video. So I don't know if you joined late, but I have a lot of papers that I referred to and showed in this very video right now. I also have a lot of videos on my channel that do the same thing, where I refer to specific peer-reviewed journal papers by evolutionists, reviewed by evolutionists, written for evolutionists. So I do cite that in my channel also. I also, as Rebecca pointed out, I also have peer-reviewed papers of my own where I refer to a lot of the literature, a lot of the evolution literature. I also have 3 books that I've written where I also discuss at length the literature. So actually, I realize you don't know who I am and you haven't looked at my work, but no, it's not true that I dance around. I'm not dancing around. I'm not citing specific study or data. Actually, I do cite specific studies and specific data. So you may have joined this video late, but that's not that doesn't characterize my work.
Awesome. Thank you. Okay. Did you have any final thoughts?
I think the whole ERV topic, yeah, I do have a final thought. The whole ERV topic is very interesting, and I don't understand it all. So I do know that argument that is exemplified by the video, that stated clearly video, is bogus. I do know that that's clearly a bogus argument, but I don't have all the answers here. It is still a lot of research. It's an ongoing research topic. A lot has yet to be done on this. Papers are coming out, just 2025, right? Yeah, we get a lot of really good papers on this coming out from people who are researching this. I plan to study it more. I plan to research it more. I have been researching it quite a bit recently. Both at a philosophical literature level, but also at the sequence data itself. I've been doing a lot of studying of the sequence data. I think it's really interesting. I think we will learn more about it. I think there'll be more light shed on it. It'll become, like all other evidences, it'll become increasingly clear just exactly how it works and how it does it. We're probably going to see that it falsifies evolution even worse than we already see. But so I don't mean to be claiming in this video that I know everything about it, or this is case closed or anything like that. But I do think it's important for us to point out that this argument is a it's a failure. The argument has failed, and that it's exemplified by the video. Is not a good argument for this truth of evolution of all these zeros. It's impressive. It's a very highly produced video. It looks really good, and when you see that huge number, those huge numbers thrown around, it's very influential. But it's presenting you with only a little bit of the information. There's a lot of information that is not being presented that needs to be understood. So that's why I think, I wanted to talk about this here. So that's my final thought.
Awesome. Thank you. And thank you to the audience, and appreciate all your questions and comments. And, if you have questions specifically about this, you can put them in the description of in the comment section. And if it's really a good topic, we can discuss, we can answer your question in a future video. So thanks for watching, and thanks, Dr. Hunter.
Thank you, Rebecca.